Hello, welcome. I'm gonna be explaining a little bit more about the painted lady experiment that we went through. So technically, I just want to start off by just explaining a little bit more. So this experiment focuses on having variable living temperatures affect the growth rate of the Vanessa cardui, in which 4 cardui larvae are split into an experimental group and a control group, in which the 2 larvae in the experimental group are grown at the incubator at 27 Celsius, while the control group are grown at room temperature, in which the different groups are grown in separate containers and are given the same type of amount of food and allowed to undergo different stages of metamorphosis. If the Vanessa cardui larvae are grown into an incubator, then they will experience faster metamorphosis than Vanessa cardui grown at room temperature. The experimental group larvae experienced better developmental compared to the control larvae and consumed greater amounts of food as well. Understanding this allows to show how warmer temperatures can affect the overall process of metamorphosis in the Vanessa cardui. So I'm going to go ahead and go more into depth about our experiment in the next following slides. Okay, so now I'm going to go ahead and go into depth about the introduction. So let me just start off by giving you guys a little bit more of descriptive, in which insects are one of the diverse species that are currently on the planet today, and they have a unique way of developing and maturing over time. This occurs throughout metamorphosis, where the insect goes from an egg to a larva to a pupa and finally reaches into a mature adult form, in which would be technically considered a butterfly. In many environmental and biological factors, they must be accounted for, liking, light exposure, amount of food given, type of food, and any other contributions to properly develop. So in particular for the Vanessa cardui, they are shown to advance in metamorphosis faster in higher temperatures compared to others at lower temperatures, and I quote Park et al. This could be attributed to the fact that these species of insects prefer to live in habitats that meet the nutritional requirements they have, and highly environmental temperatures leads to diversity of different plants and food sources compared to colder temperatures. I quote Jans 2005. However, if temperatures increase higher than butterflies, they are accustomed to it than in butterflies, so they spoil their food and thus affect their later stages of development. The Vanessa cardui are accustomed to eating a variety of different plants like calendula, mallows, sunflowers, and others to receive proper nutrients. On the other hand, artificial food that has all the nutritional values and requirements found in preferred plants, and in some cases has caused quicker growth and made them progress to metamorphosis faster, in which we're trying to explain in our experiment ourselves. And additionally, increased temperatures have allowed for the Vanessa cardui to increase how often they fly and promote migratory patterns across different regions and increasing the prevalence of laying more eggs. So based on this, if Vanessa cardui are grown into a higher temperature environment, then faster progression of metamorphosis will occur, in which we actually did receive a good experiment, and we'll go ahead and emphasize that a little bit more towards the end. Okay, so now I'm gonna go ahead and going into methods. We were given the chance to make our own experiment, which all of ours will be different. So within ours, the method we were able to use is to give the larvae about 5 grams of food per day, as we received the larvae to each. We received 4 larvae, in which 2 were going to be on the experimental method, and the other 2 will be for the control, just to determine between the changes of both. As we go on about this experiment, we determine and notate the changes between each day. Based on the limitation of our group, we were not able to gather information for one day of the week, which is the second day of the experiment, and which later on, it didn't really cause any effect just due to how much food we were giving them at that time. But as we kept gathering information throughout the week, we were able to observe the differences and changes of these larvae individually. We were still documenting the observations while we were aware of the differences of each larvae to go on about their daily growth with differences with feeding and the light of the experience. So, like I did mention earlier, since we split the 4 Vanessa cardui into 2, with the experimental group and the group, there were 2 larvae grown into the incubator, which is what I mentioned earlier with the 27 Celsius, while the group were grown at room temperature. So within the control group, they were just technically grown naturally, while we did an experimental group to see if temperature does really affect their growth. And that is technically our experiment in how we wanted to do it ourselves. Within this, we were also to experience how much the food also cost them, and if that also made an effect in how big and how they weighted later on in the experiment. At first, they were able to not weight nothing. They were still little babies as they were in their little larvae, and the food also did cost, in which is going to be more explained to Deb about in the results. Okay, so as you can see, we did add on some photographs to kind of have a visual look of our experiment. On the left side, you can see that we do have the picture of the control. Keep in mind that this is the third day of the experiment already. We were not able to gather photographs within the first day or the second day just due to limitations of our group, since there's only 2 of us. But within our control, you can see that on the left side, they're still growing naturally, but you can see that there is plenty of food for them, whether it was one day that we did skip as well as for the experimental. You can tell that they both look the same and still both have enough food for the rest of the days that were given to them. As you can tell in the experimental, I had added an extra picture to show that there was already one of the little babies already curled up, while the other ones were still kind of moving around. Within this, there was some observations that they were moving slow while the other ones were moving fast. So in the experimental ones, one of them was moving faster than the other ones, but as you can tell later on the results, that there will be the reasoning why, which would be the temperature. But as you can tell that there is 2 different types of larvae within these control and experimental. It's not really much of a difference, but later on you will see the difference between the 2. Okay,
So our results show that the experimental group grew faster and completed metamorphosis compared to the control group because of the slight increase in temperature. So the experimental group grew from 12.5 millimeters and 10 millimeters to 30 millimeters and 25 millimeters, while the control group grew from 10 millimeters and 7.5 millimeters to 25 and 27 millimeters. But the experimental group finished 2 days before the control group, and this could be indicated by the amount of food they were consuming per day. As you'll see in the next slide, the experimental group steadily increased the amount of food it ate, and the control group took a while to do that. So as a result, the experimental group reached the pupa stage of metamorphosis faster than the control group, meaning that the experimental group will soon reach its final form of metamorphosis than the control group. So here we have a data table and graph that go along with the results that I mentioned earlier. The data table and the graph, they serve to show the overall progression of the 2 larvae in each group over the course of 2 weeks. So from the data table on the left, we see the experimental group and the control groups both are given the same amount of food and are similar in length. Then after a period of time, the experimental group begins to consume its food much faster than the control group, until one of the larvae in the experimental group turns to a cocoon and is in the pupa state already. And then soon after, the other larvae in the experimental group begins to turn into a cocoon. And then the control group is taking a while to progress into a cocoon, and it isn't shown on the data table or graph here. So on the graph, we see steady improvement in the experimental groups, and as well as the control groups. It's just taking much longer for the control groups to progress through metamorphosis than it is for the experimental group. So here we have a data table and graph that go along. So here we have a data table. So as mentioned previously, the experimental group was grown in an incubator of 27 degrees Celsius and was grown there throughout the entirety of the experiment up until they were being fed or weighed, while the control group was grown under 12 hours of light and 12 hours of darkness. So We see in these two pictures that the experimental group, by the time one of them turns into a cocoon, there's not that much food, indicating they complete metamorphosis faster, while in the control group, they were taking a little longer. So as mentioned previously, the experimental. So at the end of our experiment, it was determined that our hypothesis was correct in predicting that the experimental group, which was grown under slightly higher temperature than the control group, would result in a faster metamorphosis of the larvae. And then understanding this can allow us to see how certain organisms react to certain living environments. For example, if we grow butterflies in a slightly higher temperature region than a cold region, this can affect how they lay eggs, where they migrate to, their ability to fly, which in turn affects things like pollination and plant production, which is very important to the environment and to all life forms that live in that area, and contribute to the overall ecosystem. So at the end of our experiment, it was determined that. So at the end of our hypothesis was correct in predicting that the experimental group, which was grown under slightly higher temperature than the control group, would result in a faster metamorphosis of the larvae. And then understanding this can allow us to see how certain organisms react to certain living environments. For example, if we grow butterflies in a slightly higher temperature region than a cold region, this can affect how they lay eggs, where they migrate to, their ability to fly, which in turn affects things like pollination and plant production, which is very important to the environment and to all life forms that live in that area, and contribute to the overall ecosystem.